Hello everyone, this is Anwar and welcome to the second lecture in our physiology video series titled Introduction to Cellular Physiology. This is in continuation to the first introduction lecture and will discuss briefly the structures and functions of various components of the cell. So to start off, let us look at the structure of the cell. This is a schematic showing the structure of the cell as seen under a light microscope. From the schematic itself, it is evident that the cell has two major components. One is the nucleus and second is the cytoplasm. The nucleus and the cytoplasm are divided by the nuclear membrane, whereas the cytoplasm is divided from the extracellular fluid by the cell membrane. The cytoplasm and nucleus contain various components which together form the protoplasm of the cell. So all the components of the cell together are known as the protoplasm of the cell, and this protoplasm consists of proteins, nucleic acids, carbohydrates, lipids, water, and various ions. So let us go through the various constituents of the cell one by one. The first and the foremost is water. Intracellular fluid consists of 70 to 80% water. So a huge amount, or the majority amount of constituent is water. So the various chemicals are dissolved in the water itself, and the organelles of the cell are present as suspended particles in the intracellular fluid. Biochemical reactions occur at the surface of these suspended particles or between dissolved components of the intracellular fluid. Next, we move on to ions present in the intracellular fluid. So the ions are the inorganic chemicals for the cellular reactions and are very necessary for various cellular machinery. So all the biochemical reactions that occur inside the cell require various ions, which in many cases act as cofactors, and most or many of the reactions inside the cell cannot proceed without the presence of particular ions. So for example, DNA replication absolutely requires the magnesium ion. So without magnesium ion, DNA replication cannot proceed. So the ions are divided into two major parts: the ions which are present in high concentration in the intracellular fluid and the ions which are present in low concentration. High concentration ions present in high concentration in the intracellular fluid include potassium, magnesium, phosphate, sulfate, and bicarbonate ion. Sodium chloride and calcium ions are present in low concentration in the intracellular fluid. Next, we move on to proteins. Proteins are the most abundant component of the cell after water and consist of 10 to 20% of the cellular mass. Proteins are divided into two major halves: the structural proteins and the functional proteins. The structural proteins are essentially filamentous structures which are polymers of individual protein molecules themselves. These structural proteins form various structures such as the microtubules and play a supportive role to the cell in the form of the cytoskeleton of the cell, as well are present in the extracellular matrix where they support the external structure of the cell. Next, we move on to the functional proteins. Functional proteins are essentially the biocatalysts and the enzymes that take part in various biochemical reactions. The enzymes, the functional proteins must come into direct contact with various substrates in order to catalyze the specific intracellular chemical reactions. Next, we move on to lipids. Lipids are several substances that are grouped together because they are soluble in fat. Lipids are essentially organic compounds which are fatty, oily, or waxy in nature and consist of 2% of the cell mass. The most important lipids include phospholipids and cholesterol, which form the cell membrane of the cell. Most of the lipids are essentially made of hydrocarbons, are mostly hydrophobic in nature. Lipids have huge use in the cell and act as signaling molecules, structural materials of the cell membrane, as well as energy reservoirs for the cell. Next, we move on to carbohydrates. Carbohydrates are sugar molecules which contain carbon, hydrogen, and oxygen, and are the major source of energy and nutrition in the cell. Our body does not store huge amounts of carbohydrates. And in normal cell, the carbohydrates only are only contribute 1% to the cellular mass. But this can increase in case of muscle cells, where the carbohydrates constitute 3%, and in liver cells, where the carbohydrates contribute 6% to the cellular mass. But carbohydrates in the form of glucose are always available in the bloodstream and are stored in the form of glycogen in the liver. These next, let us move on to nucleotides and nucleic acids. Nucleotides are organic compounds which are consisting of a sugar, phosphate, and a base, which are linked together by various chemical bonds. Nucleotides are building blocks of nucleic acids joined by phosphate groups via the phosphoester linkage. So nucleotides join together one after one after one in order to form the nucleic acids. There is a particular directionality to how the nucleotides are joined, and nucleic acids always join together to form the RNA and DNA of the cell from the 5' to the 3' direction. In case of RNA, the particular sugar moiety involved is a ribose sugar, but in case of the DNA, the sugar is a deoxyribose sugar. Nucleotides are also present in the form of ATP or adenosine triphosphate, which is the energy currency of the cell. Next, here we have an enlarged picture of the cell itself. And here we can see various structures of the cell. These include the nucleus, and which also has the nuclear membrane, the endoplasmic reticulum, the Golgi apparatus, mitochondria, lysosomes, ribosomes, microtubules, secretory granules, all which constitute the whole cell. Here we can also see the chromosomes as well as the DNA inside the nucleus. Next, we move on to various structures of the cell and discuss them in brief, and also discuss in brief their functions as well. So first off, we have the cell membrane. The cell membrane is a thin double-layered film of phospholipids that cover the entire cell. The cell membrane, which envelopes the entire cell, is a thin, pliable, elastic structure only 7.5 to 10 nanometers thick. It is composed entirely of lipids as well as proteins. The approximate composition of the proteins is 55%, phospholipid 25%, cholesterol and other lipids are around 17%, and finally, carbohydrates are also present in the cell membrane, which contribute 3% to the total composition of the cell membrane. The structure is a lipid bilayer. So what do we mean by a lipid bilayer? So essentially, the cell membrane is composed of phospholipids. Phospholipids are consist of two parts. One part is the hydrocarbon chain, which is present, and it is hydrophobic in nature, so it does not like water. And on top of it sits a phosphate group, which is hydrophilic in nature, which likes water. So these are present back to back. So which means is that the hydrophobic regions of two phospholipids are present in the interior of the cell membrane, and the hydrophilic region, that is the phosphate groups, are present on the outside. We have to understand that the extracellular fluid as well as the intracellular fluid both contains huge amount of water. So the phosphate groups are happy in being present on both ends of the cell membrane, whereas the fat soluble hydrophobic interior is made up of the hydrocarbon backbone. So the whole structure is completely impenetrable to water molecules, as there is obviously a layer of fat in between, so water molecules cannot penetrate. But water molecules of water soluble substances also cannot penetrate inside the cell membrane and pass into the cell. But fat soluble ones can easily penetrate through the cell membrane and come into the cell itself. Next, the cell membrane also consists of a huge amount of proteins, which include the integral proteins and the peripheral proteins. Integral proteins are proteins which pass right through the cell membrane. So they are present on, they have parts which are present intracellularly as well as extracellularly also. These include various protein channels, carrier proteins, and receptors. Next, there are peripheral proteins, which are like this. These do not go right through the entire cell membrane, but are present at one end and are mostly attached to integral proteins and act as enzymes or signaling molecules. The cell membrane also consists of a huge amount of carbohydrates present on the cell membrane on the extracellular surface of the cell membrane itself. These carbohydrates are present attached to lipids and proteins in the form of glycolipids or glycoproteins. So, what is a glycolipid? So, glycolipids are lipid molecules which with small amount of glyco or sugar moieties attached to it. The whole cell membrane consists of a huge amount of sugar molecules present on its surface, and this is known as the cell glycocalyx. This cell glycocalyx has a number of functions, which include giving a negative charge to the cell membrane, as well as helping for attachment of the cell to various surfaces. And finally, the cell glycocalyx acts as receptor sites for various ligands. Next, we move on to the nucleus of the cell. The nucleus of the cell is essentially the control center of the cell, and it contains DNA, which stores information or the hereditary information in the form of genes. During cell division, DNA is the first molecule to be divided, and it forms two identical sets. If this process of forming two identical sets, which is known as replication, does not occur properly, then the cell does not go further for cell division. The nucleus is, as we discussed, separated from the cytoplasm via the nuclear membrane, which allows only selective entry and exit through nuclear pores present on the nuclear membrane surface. So these are guarded by nuclear pore complexes that do not allow most proteins or other molecules inside or out from the nucleus without special signals present, which are known as nuclear localization signals. Next, we move on to the endoplasmic reticulum. Endoplasmic reticulum is a network of tubular and flat structures which are folded in nature. The folding causes the ER to have a surface area of 30 to 40 times larger as compared to the cell membrane, and the contains endoplasmic matrix inside it. ER is the site of synthesis of the proteins and lipids present in the cell. The proteins are synthesized in the granular or the rough ER, and the smooth ER is responsible for the manufacture of various lipids in the cell. The rough ER has a rough or granular structure due to presence of embedded ribosomes on top of it, which are the protein manufacturing machinery of the cell. And the smooth ER does not have any ribosome attached, and thus we have a granular structure or topography of the smooth ER. Next, we look at the Golgi apparatus. Golgi apparatus is closely linked to the endoplasmic reticulum and is known as the packaging center of the cell. Golgi is responsible for packaging various cargoes and then sending them to the appropriate locations. The membrane of the Golgi apparatus is similar to that of the smooth ER. Various transport vesicles carry the different cargoes which are produced in the ER, which includes proteins as well as lipids, and then transport them to the Golgi, from where they are transported to various parts of the cell. The Golgi apparatus processes these cargoes and sends them to their appropriate locations, which can also include extracellular proteins, which are secreted out of the cell via secretory vesicles. In case the Golgi encounters improperly folded proteins or proteins which are non-functional in nature, the Golgi then directs these proteins to lysosomes, which are degradation complexes, which will completely degrade the proteins themselves. So essentially, the Golgi is the packaging center of the cell, which is responsible for the localization of various cargoes to different parts of the cell. Next, we move on to mitochondria. The mitochondria is obviously the powerhouse of the cell and is present in all areas of each cell and is responsible for major share of the energy metabolism which is present in the cell. The mitochondria is divided, has two lipid membranes, the inner mitochondrial membrane and the outer mitochondrial membrane. The mitochondria is unique in the fact that mitochondria can autonomously divide and also contain mitochondrial DNA, which also autonomously replicate. The inner matrix of the mitochondria consists of various enzymes which produce the energy currency of the cell, ATP. Now let us look at various other organelles present in the cell. The first one we are going to look at is the lysosome. Lysosome is the intracellular digestive system of the cell, which digests or degrades various particles. It is full of hydrolase enzymes which can break down proteins, glycogens, as well as lipids. So any molecule that comes into contact with the lysosome, which will be completely degraded. So how is the lysosome useful for the cell? The lysosome helps in digestion or degradation of various damaged cellular structures, digestion of food particles, and degradation of foreign unwanted matter. Next, we move on to peroxisms. Peroxisms are similar to lysosome in which they are also single membrane structures, but instead of hydrolases, they contain oxidases and also can self-replicate. So, what is the function of the peroxisome? Peroxisome contains various oxidases which deal with various reactive oxygen species or ROS, such as hydrogen peroxides. ROS are very damaging molecules that are created during normal cellular respiration, which include hydrogen peroxidases and superoxides. These can react with various proteins, lipids, and nucleic acid present in the cell and degrade them and destroy them. And thus, these need to be dealt with. And the peroxisome is responsible for dealing with these ROS. Finally, we come to secretory vesicles. Secretory vesicles are closely related to the ER-Golgi complex, as we discussed before, and most vesicles are created by the ER-Golgi complex. These are storehouses of protein, lipids, and other cargo, which are then secreted from the cell. Next, we have a comparison chart of various organisms. The first we have is a small virus, which is only 15 nanometer in length, followed by a large virus, which are around 150 nanometer in length, followed by Rickettsia, which is a bacterium, and an intracellular parasite, which is around 350 nanometer in length, and a normal bacterium is around 1 micrometer in length. And finally, we have a human cell, which is around generally 5 to 10 micrometer in length, but human cells can also can obviously be much larger or smaller than the general picture that is presented here. Next, let us look at various functions performed by the cell. The first we want to look at is pinocytosis. As we discussed before, the cell membrane is impenetrable to water-soluble molecules, and fat-soluble molecules can enter, but large fat-soluble molecules are also have difficulty in entering through the cell membrane directly. Thus, the cell requires a particular process via which large macromolecules can enter through the cell membrane, and this process is known as pinocytosis. So, how does pinocytosis occur? The macromolecule which wants to enter into the cell first it has to be directed towards the clathrin coated pit. This pit contains various receptors for these molecules. Once the molecules attach to the various receptor proteins, the coated pit is activated, after which clathrin, actin, and myosin cause invagination of the pit. So the pit starts folding inside and the border starts to close. Once the borders join together, the clathrin starts dissolving and actin pulls the vesicle away from the cell membrane. And thus we get a pinocytotic vesicle form, which now has the particular proteins inside the cell. Next, we look at phagocytosis. Phagocytosis is the process by which the cell ingests large particles, as large as other cells, instead of small molecules. This process is done mostly by macrophages and blood cells. In this process, the receptor attaches to the receptors present on the macrophage attach to the particle link. Presence of antibodies on the foreign particle itself also helps in attachment of macrophages to the also helps in attachment of the macrophages to the foreign particle, and also improve the recognition of the foreign particle by the macrophages. This process by which antibodies make a foreign particle more tasty to the macrophages themselves is known as opsonization, and we will look at opsonization in detail in later lectures. Once the macrophage receptors are attached to the particle ligands, the edges of the membrane of the macrophage envelop the particle in a zipper-like action. Actin then contracts to push the phagocytic vesicle into the cell itself, and contractile proteins pinch the vesicle off. So we now have a vesicle formed inside the cell that contains this foreign particle inside it. Now what happens is that this phagocytic particle moves into the cell where lysosomes are targeted towards it. And as we learned, lysosomes are degrading complexes. This phagocytic vesicle or phagosome then combines with the lysosome to form the digestive vesicle or phagolysosome. The degrading enzyme present in the lysosomes themselves then degrade these foreign particles or other particles that were ingested by the cell, and the residual body, which contains neutralized particles as well as degraded particles, are then excreted by the cell. Next, let us look at the functions of the Golgi complex. So the protein synthesis of the cell is done by ribosomes, which are attached to the rough ER. The proteins, as they synthesize the new peptide, transport the peptide right into the endoplasmic matrix. Inside the endoplasmic matrix, the protein undergoes folding as well as post-translational modifications such as glycosylation. And after the protein has been correctly folded and undergone various post-translational modification, it is targeted to the Golgi. In the Golgi, the protein is checked for correct folding and functionality of the protein. And if the Golgi finds any particular protein to be non-functional or incorrectly folded, the protein is then directed towards lysosomes where it is degraded. Otherwise, if the protein is correctly folded as well as is functional, the Golgi then sends the particular protein via transport vesicles to various parts of the body. Or in case the protein needs to be secreted out of the cell, then it is transported to the cell membrane via secretory vesicle, from where it is secreted out of the cell. After this, let us look at the functions of the mitochondria. The mitochondria is obviously the powerhouse of the cell and is where cellular respiration occurs. Glycolysis always occurs in the cytoplasm, where glucose is converted to pyruvic acid while producing two molecules of ATP from ADP, a process known as substrate level phosphorylation. This pyruvic acid is then converted into acetyl-CoA, which is transported into the mitochondria itself, where the oxidative phosphorylation reaction occurs, which creates a tremendous amount of energy in the form of 36 ATP. Thus, the process of cellular respiration is the major component of the energy metabolism of the cell and produces a huge amount of ATP, and the whole of the cellular respiration occurs inside the mitochondria itself. Here is a schematic and a schematic of the reaction that actually occurs, overall reaction that actually occurs. Next, we look at locomotion in cells. For movement of the entire cell, the cell undergoes a process known as amoeboid movement. In amoeboid movement, the cell projects pseudopodia, which are essentially projections of the cytoplasm to the new region the cell wants to move into. Once the pseudopodia reach the new location, it attaches to the extracellular matrix there and pulls the entire cell to the new position. A great example of amoeboid movement is performed by white blood cells as they move to sites of infection in a particular tissue. The stimulus for white blood cell movement are chemotactic agents released by the various cells of the immune system, and the whole process is known as chemotaxis. Next, we move on to cilia and ciliary movement. Cilia are hair-like structures present on the surface of some cells and produce a whip-like movement, which looks something like this. The forward stroke and the backward stroke look something like this, and are responsible for moving particles along with them while they move. For the ciliary movement, ATP is hydrolyzed to ADP, that is, energy is used. These cilia are present in respiratory tracts and the uterine tubes in the reproductive tracts. In respiratory tracts, they are used to move foreign particles, and in the uterine or the fallopian tubes, they are used to move the egg cell down the fallopian tubes. Finally, this is the reference for this particular lecture, which is the chapter 2 of textbook of medical physiology by Arthur C. Guyton, the 11th edition. Uh, any comments or any questions about the lecture are more than welcome, and you can put them down under the video themselves. Thank you for watching the video.